Thank you for inviting me to come down. I was in San Diego for 6 years at UCSD, and I left about 10 years ago, and my wife is still not happy with me, nor my kids. And every time I come down here, I'm like, oh my God, how could I leave? San Diego is an amazing place. But I still come down once a month. I have so many collaborators and a couple of companies and stuff in San Diego, so it keeps me down here. So, a lot of science is, you start from the basic science and you think about a disease and then you look at it and then you come up with an idea and then you go to the clinic, which is a very long process. But sometimes, we can get lucky, and that's what happened to me in my journey of getting into chordoma research and into the chordoma space. And I'll show you how I got into it. I have a lot of conflicts. So there's basic science to the clinic, which takes a long time, but there's also learning from every single patient in clinic and applying and coming up with an idea based on what we see in the clinic and applying that to the patient. Because right now, there are, in the last 10, 15 years, about 1,000 new cancer drugs approved. And there's no way we will ever have the opportunity to do 1,000 clinical trials in chordoma fast enough. And then when you think about the combinations, we'll never get there, right? So we have to sometimes, obviously the science could help us figure out personalized medicine, but sometimes you get lucky and then you have to follow up on that as well. So you guys know all of this already, but it's a rare tumor. No really officially approved drugs. There are drugs that we use. And I'll start off with this case of a 55-year-old who had a metastatic clival chordoma, failed all of the standard surgeries, radiation, and a bunch of drugs as shown there, including immunotherapy. And he came to me because he had lesions in the brain itself, metastatic to the brain, which is very unusual. And at the time, as a neuro-oncologist, I think about getting a drug into the brain, how we can do that better. And Pemetrexed is a broad anti-cancer drug. It's approved for lung cancer, but it actually works in many, many different cancers. And at the standard dose of 500 milligrams per meter squared, it's not enough to get into the brain. And we've used it for lymphoma, breast cancer in the brain, at a higher dose, 900 milligrams per meter squared, and actually even you can go up even higher than that. But at that dose, it's a pretty reasonable level. And when we did that for this particular patient, as you can see here, before and after, the tumors in the brain shrank. The tumor in the retroperitoneum in the back also shrank as well, quite dramatically within a few cycles of treatment. And so then I said, well, why did this patient respond? And we looked at a database with Caris, which is a profiling company. They do all the genetic markers. We use them quite a bit. And in their database, one of the markers of response to Pemetrexed that could help us personalize medicine is called TS, thymidylate synthetase. And in their database, only 16%, I think that's right, it says 16% are positive. And actually positive means you won't respond to Pemetrexed. So most patients, 84%, are negative, so there's a high chance of response to Pemetrexed. The bottom line is that patient, as well as another patient, had very low TS, thymidylate synthetase, which is at the bottom row there. And another patient that we got from a colleague in Toronto who treated a pediatric patient, based on our data, he said, let me try it on a pediatric patient. This was 5 years ago, by the way. And he did treat the patient. Unfortunately, the tumor grew and didn't respond. And that's on the bottom right there. So that patient had a high percentage of TS, 16%, whereas the other two patients where their tumor shrank in the lung and in the brain had a low percentage. So it's a small sample size, but this is exciting that we can maybe select the patients who are more likely going to respond to this drug. So this is the data that I showed the Chordoma Foundation 4 years ago, before COVID, and that actually got them to fund a clinical trial in a formal prospective way, because these treatments were all off-label treatments. I just got the drug approved for my patient and just treated them. So basically the rationale is that Pemetrexed is this old chemotherapy that inhibits DNA synthesis, which many chemotherapies do, but it does it in a very broad way. And when we use the higher dose, it gets into the brain very well as the rest of the body. And we know that 84% of patients or chordomas may respond to it based on the marker that I mentioned, and we already showed that we had 2 patients that responded, which was the basis for the approval, for the clinical trial approval from the FDA as well as from the Chordoma Foundation funded it. And that's shown here. So we treated 15 patients, and this was during COVID. It took about 1.5 years, which is actually pretty fast if you think about it, because we literally enrolled the first patient in April 2020, I think. And so we had 8 clival, 4 spinal, and then 3 sacral chordomas. Most of the patients had, a third of the patients had prior treatment, prior drug treatment. 80% of the patients had prior radiation, and you can see the rest of the mix of the tumors. About a third of them were metastatic, meaning outside of the main area, into the lungs or liver or to the skull. And we had a reasonable response rate. There was, as you can show, this is what's called a waterfall. So the tumors at baseline compared to after treatment. You can see on the far right the tumors that shrank during the treatment, and then on the left the tumors that increased during the treatment. What was really remarkable is this one patient from Ohio who had complete blindness in the right eye because the tumor was pressing on the nerve coming out of the eye. And that person actually, in 3 months, noted improvement and the vision improved, and on the imaging, the tumor shrank in the bone. So that was pretty exciting for us to see clinical benefit. And even though we had really 2 remarkable responses on the study with shrinkage, objective responses, a lot of the patients had symptoms, pain or nerve pain or whatever it was, and a lot of patients' symptoms improved, even though we couldn't see. These tumors are very hard compared to other tumors of seeing them shrink because the bone is there and bone doesn't shrink. So it is a challenge for us sometimes to know what's happening. But clearly, we had benefit on imaging as well as on many patients had symptomatic benefit with various symptoms that they had. So this is the summary of the study. The toxicity was pretty manageable. There was a 14% partial response rate, 71% stable disease, which means over 85% benefit rate. The progression-free survival was 10 months, and this is comparable or better than other studies that have been done in the past. And then, as I mentioned, that TS testing, we did it in this cohort. Unfortunately, because it's only 15 patients, it's hard to see if it correlated, but it did correlate, but not statistically significant. So patients who had low expression did better than patients who had expression. And we did a bunch of other fancy stuff. We looked at the blood. Can we diagnose chordoma in the blood? Like a PSA, for prostate cancer, you can monitor. So there's these microRNAs that we've used, and we were able to, on the left is red, is all the chordoma patients' blood, looking for microRNAs. On the right is normal patients' blood, and clearly there's a difference in the blood. And we use that to identify chordoma versus non-chordoma. But because of the small number of patients, we couldn't tell whether the levels over time, we were using it and monitoring patients over time, whether the levels correlated with response because it was such a small sample size. We hope to explore this in the future, but it's good that we have something that we can look at that we can validate in the future. So that's what really started our journey into chordoma, and now what can we do to improve the chances of response? And in much of oncology, as you guys know, is the immune system. This is blown up in the last 10 years from melanoma to lung cancer to breast cancer to every other cancer. And so it's natural to ask, can we use the immune system to improve upon this? And the immune system, the reason you have cancer is because the immune system doesn't recognize the cancer and doesn't know how to get rid of it. And usually the tumor expresses things like PD-1 CTLA4, and a bunch of other proteins that basically cloak the cancer, and the immune system doesn't recognize it. It blocks the immune system. So now there are drugs, there's about 20, 30 drugs that block this blocker. They're called PD-1 inhibitors or CTLA inhibitors, and one of them is called pembrolizumab or Keytruda, which is approved for like 20 different cancers now. It's an amazing drug. So we decided to combine that with the Pemetrexed that we showed had activity to see if we can further improve the response rate. And this is the new study that actually just got funded by the Chordoma Foundation, and we officially opened it this past week. We got all the approvals, so there'll be an announcement sent out about it. But I just want to go over the rationale for this. So we already showed that Pemetrexed on its own is active. That was the data I showed earlier, about 20% say response. The PD-1, the marker for Keytruda, is expressed in 50% of Chordoma patients. And there was a study that was published last year that included all sarcomas, but there was about 34 patients with Chordoma that were treated with just pembrolizumab alone, and it wasn't a very good response. It was like a 6% response rate. So it's a very low response rate. Now, does that mean it doesn't work on its own? Yes. But what we learned about immunotherapy is sometimes when you combine it with other things like radiation or chemo, you can get an improved response. And so the rationale for combining them is really important, and that's the fourth point, that when you combine a chemo with the immune therapy, the chemo kills the cancer and then it makes it recognizable by the immune system. And so that's why the rationale is very good for combining, and maybe the immunotherapy would boost the response to Pemetrexed even more. And so this is what's called a phase 2 study. We're looking for safety and efficacy of the combination of both drugs. We'll collect the tumor tissue at the time, the previously collected tumor tissue, and then blood throughout the treatment, and see if we can correlate who responds to the markers that I told you about earlier. So this is a pretty standard, we're looking for objective response rate of the combination, and we know what the comparison is for the two other drugs. So is the combo going to be better than those two other drugs? And then there's a whole bunch of other endpoints, overall survival, progression-free survival, and quality of life. As I mentioned in the first study, we saw really dramatic benefit to some people, even though we couldn't see the tumors shrink because they're tricky sometimes, but clinically patients had a benefit, felt better, less pain, things like that. And obviously we want to also look at blood and see if we can track the disease with those markers I mentioned earlier. So the main inclusion for this study is to have a diagnosis of chordoma, have progressive disease, meaning we have to have evidence that it's growing over time, and be able to measure it. There's a whole bunch of other boring criteria as well, and we have to make sure their blood work is good, the labs are good, the liver is functioning fine, the kidneys functioning fine, there's no infections and things like that. So it's pretty standard for all of our trials.
Can I ask one question? So for the inclusion criteria, can you just include the TS?
So we decided not to do that because when you start combining drugs, sometimes it's not, it may not hold. I think it's still a good marker, but it's a gradation of immunohistochemistry. We will be testing for it, but I don't want to bias it against that. Does that make sense?
Yeah, I still think, if you find the correlation between TS and the chemo agent, you should do it because, this is still very toxic. So in terms of chordoma, I don't know how many T cells that, it's like a cold tumor or hot tumor.
Yeah, so you raise a very good point. So I don't have data yet to say, there's a correlation I feel is real, but I don't have data to confirm it because the sample size is too small.
Yeah.
So I can't say don't treat these patients because they have TS positive, because it's not true. I haven't proven it. So we're not going to use that as an inclusion criteria at this point. Now, There are patients getting Pemetrexed. I treated a few other patients with Pemetrexed off trial, and my colleagues have also treated, and I just heard a few patients internationally getting treated. If we gather all that data and add to that data set of the TS marker, is it good, is it predictive or not, then we can be more comfortable. Even after that, even if I statistically prove that TS matters, sometimes when you do combinations, you could get a better response, and it may not matter after that, if you understand what I'm saying.
Yeah, so at least from biopsy samples, can you say tell whether the T cells in the tumor? If there's no T cells, it's very hard to get efficacy.
Are you a scientist?
Yes.
Yeah.
That's good. So, no, you're right. So, we have the sarcoma meeting across at the hotel, and in every other cancer meeting also. There are cold tumors and hot tumors. And the hot tumors respond to checkpoints, you're absolutely right, better than cold tumors. The question is, how do you make a cold tumor hot? And Pemetrexed is one of those drugs that can make a cold tumor hot by killing the cells. So while what you said is accurate, what I'm saying is when you combine drugs or do things that cause cell death, tumor cell death, you can get an immune response, okay, infiltration. And the Keytruda will help with that.
Could you explain what hot and cold?
Yeah, hot means like melanoma, they have immune cells there, T cells, and but the immune system is blocked, like PD-1 and CTLA, and so when you add just Keytruda, they respond amazingly well. So the immune system is primed, ready to go, they're just blocked. Whereas a cold tumor, there's no immune system is not even there. They don't realize that there's a tumor there, and so you have to bring the immune cells there, and you have to make it hot. Radiation can make it hot, chemotherapy can make it hot, and a whole bunch of other drugs out there that are in development can make it hot, meaning bring the immune system to the tumor. So it's a good question, but it's just hard to, in this rare population, you can only do so many studies at one time, so.
Does chordoma fall into one of those categories? Or
More cold than hot. So that's highlighted because I showed you earlier that this study, it was 6% response rate, so very low response rate to just immune therapy, which means they're cold. Many sarcomas are cold, but the chordoma one was very, very low response. Yes?
You mentioned that you have patients who are not in the study and there are other patients out there. Do you want their other patients' information?
Absolutely.
Okay.
Absolutely. We've talked about a database of collecting everyone's in the country and the world's data and putting it all together. Yeah.
My son is on it, not anything else right now. Student's
Good.
stable.
Good.
Been on it since July, but I didn't, who sends that? His oncologist, who sends that, or his surgeon, who sends that to you?
Oh, for the testing?
You said you like your data, so.
Yeah.
Should his doctors get in touch with you?
Yeah, doctor, the patient.
Okay.
I'll take it any which way I can get my hands on it. Because we really need to publish all this data, because since the publication, a lot of people have been using it. I bet you tons of patients in China, because I got a bunch of emails in India also.
Yep.
I bet a bunch of patients are getting it, so it'll be great to do some kind of announcement saying, please share your data, you know?
Yeah.
Great, that was great point. Okay, so where was I? Forget all the inclusion. We'll figure it out, once you contact us. So the Chordoma Foundation graciously funded this, but it's at a single site, which is Santa Monica. We hope in the future to open it at other sites when we get more funding as well, because it is a rare disease, and I do feel bad that patients have to travel. We had that patient that had the vision loss came from Ohio every 3 weeks. She was an amazing woman, and we need to open a multi-center in the future, and I think we will after we get started here. So the treatment is every 3 weeks. We do the combination of the two drugs I mentioned, the immunotherapy and the Pemetrexed, and it's a 21 patient study, which is for a rare tumor type. It's a good enough sample size for us to see if it's better than what we've done in the past, and if it's really active, we can then think about adding more to this or doing a larger study for FDA approval in the future.
What's the
that-
major side effect?
Yeah, I'm actually getting to that right now. So obviously we look at safety, and we'll do 3 patients, look at their safety of the combo. Now this combo is not new. This combo is a standard combo for lung cancer and probably other cancers. Pembro, Pemetrexed, and carboplatin are a standard 3 drugs for lung cancer. The only difference here is we're not doing carboplatin, so that's good, less side effects, but it's a higher dose of the Pemetrexed than the lung cancer dose. So we'll make sure it's safe, which I know it is because I've treated patients off-label already previously, so with other cancers. But it's every 21-day visit. There's a standard blood work and things that we look. What are the side effects you asked? So the Pembro, it's very well known. 20% of patients get a bunch of side effects: fatigue, rash, pain, rash, diarrhea, nausea. It's all in there. But the main things that we worry about is the immune-related side effects, the rash, diarrhea. So immunotherapy causes inflammation to kill the cancer, but it can cause side effects of inflammation in the skin, which is a rash, or the gut, the diarrhea. We know how to control all those side effects pretty well these days. So it's pretty well tolerated compared to just chemo. The Pemetrexed is a pretty low side effect chemo compared to carboplatin and other like breast cancer type of drugs, Taxol and things that really you lose your hair and all that stuff. This mainly causes fatigue. It can drop your blood counts. And the main side effect over time that it causes in some patients is swelling in the legs, which we control with the little steroids. So those are the main side effects of. There's probably more if you look at the literature and stuff, but those are the main things that we monitor for. And then, as I mentioned, as part of the study, we collect blood for research studies, looking for those same markers. There's also new companies that we routinely do, and actually I should have put that in here because I have a few chordoma patients where we could see the markers going down or up depending on whether they're responding or not. So we're going to be studying the blood to see if we can identify markers of the disease as well. And so, anyway, we are officially open. So we have a few patients lined up, and we're happy to. 21 patients. We have to treat 3 patients first, make sure they're okay for 3 weeks, and then we can enroll as many patients as we want after that. So we hope to enroll and accrue and get this study finished quickly. The other study we enrolled within 18 months, which was good. So I think we can even do it faster since the Urbextech study is about to close as well. So it's a good timing that we're getting this open. That's it. Any questions? Yes.
I thought your talk was really interesting. I had a couple questions. You had talked about possibly blood diagnosis using biomarkers, and that would be a way to diagnose chordoma. Do you think that would change the way that it's diagnosed now? Because to my understanding, I think it's through MRIs.
Yeah. So it was interesting. There was this. So we had normal blood from other just a repository, and but I think it was like these guys here may actually have chordoma that we just don't know about because they have the same profile as these guys. I am going to put together, once we get this study going, put together a proposal to, hopefully Chordoma Foundation can raise money to do it, but to collect blood on patients like that have chordoma around the country and just do the same profiling and see if we can make some correlation. But there's also new technologies now, ctDNA, not just microRNAs, and so I think it's an important thing. When you have a bone lesion, you know what it is. Can you do a blood test to diagnose it? Or the tricky part for me is, I mean, it's even in brain tumors after we do radiation, we can't tell is it tumor or radiation necrosis or inflammation. It's a very, very tricky thing. More for the brain than probably other, but the bone is also just as tough to understand what we're seeing on the imaging. Is it inflammation, tumor, necrosis, whatever? It's difficult. Yes.
So in your combination of meds, why did you try Keytruda instead of something like Gleevec, or why did you choose that one?
Good question. We're in this era of immunotherapy is one reason, and there is a lot of good data that combining, like in the lung cancer field, Pemetrexed, killing the tumor cells makes the Pembro work better, or vice versa, however you want to think about it. So the combo is very scientifically sound. And then we have here an active chemotherapy drug that can kill the cell and expose it to the immune system. What happens when you damage a cell is it reacts to it, like expressing PD-1, PD-L1, these immune markers that the Pembro and these other drugs can block. So they still try to cloak and escape the cell death, escape the immune system. So the combo makes sense to increase the response rate is the main thing. And it's not another chemo, like I did actually originally propose Pemetrexed and carboplatin because the platinums have some activity as well, very low, but so we could combine that, but then that means more toxicity, which we worry about in all patients, but in particularly for chordoma patients. So the Pembro hopefully will give that balance of improving the response rate, improving progression-free survival, overall survival, but not create more toxicity for that. So that's the idea. Yes,
You know that, sorry.
go ahead.
This may be out of your area of expertise, but do you have anything to say on the prospects for a brachyury drug?
We actually did participate in one of the trials. So it was promising. We had some patients respond, but I think everyone knows, right, it was stopped, right? We know that, right? It's public. Yeah, so at least that one vaccine approach, while promising, at least is not moving forward at this point.
Well, that's a good question for Dan this afternoon.
Okay.
Dan Freed, yeah.
I was wondering.
Yep.
All right.
Yeah.
Can I ask you a question? Say for instance, you are taking Pemetrexed off label, and it's not causing shrinkage, but it keeps it stable. Say for instance, you've been on it for a certain period of time. Would you say adding the Pembro could that be a benefit? If
Yeah, we don't know yet.
Can you repeat what you said?
Yeah, sorry. She's asking if you were already on Pemetrexed and you're stable, a year later, would you want to add Pembro or other drug, right? And I don't know the answer. I have done that. We've also done, another trial was that ADAPT study that was presented as well, which was injecting the tumors with the oncolytic virus that has a different immune blocker called TGF-beta. And when we did it alone, it didn't do much, but when I added Pembro off-label later, the tumor shrank. So, I mean, it's a case-by-case situation for any particular patient about adding another drug. Is stable good or not good? Was it growing before it's complicated? And do you want to add the toxicities or not? But it's something to do, for sure.
Yeah, so chordoma is not sensitive to chemo agents. Like for this, if say the tumor cannot respond to Pemetrexed, and how can you expect people will have some effect?
Yes and no, because there is a population. Oh, sorry. She said if chordomas don't respond to chemotherapy, how do you expect it to respond to immunotherapy? Yes, but no, because I showed you data that it does respond some patients. So the other tricky part is we call it chordoma, but every single patient is very different. And so there's subsets within subsets. So TS is one marker, but there's EGFR. There's I have a patient right now who has an IDH mutation, chordoma, that's on an IDH inhibitor, and clinically she's improving. So there will be a lot of personalized medicine that needs to be involved here. But I'm hoping, I mean, there was 2/3 of the patient, there was slight shrinkage to really good shrinkage. And the other big marker, I think, is actually how aggressive it is. The faster you grow, the more sensitive you are to chemotherapy, right? So I think in our patient study, we probably had more aggressive tumors because we had to have documented growth before we enrolled in the study. We didn't want to take stable patients because it's hard to know whether it's helping. So I think we did select for that, and I'm sure that's part of the response is faster growing, more response than slower growing. Yeah.
I wonder if you've done any studies or haven't written any studies done looking at Pemetrexed and its induction or possible induction of HLA class 1 expression. You know, because that would answer the question of how you might make it more susceptible to pembro. And just like HDAC inhibitors can sometimes induce HLA class 1 or radiation. Maybe that might help provide a mechanism for that.
Absolutely. And there is data from the lung cancer literature about it. For Chordoma, we could do a study with the Chordoma group, but we also did Temodar, actually temozolomide, since I'm a brain guy. Temodar gets into the brain, and no one's ever done Temodar for Chordoma. So in the preclinical models, which we published last year, that the Chordoma Foundation did, Dan did actually, and the previous people, Temodar had some activity, and the Temodar plus Pemetrexed has activity as well. But we can look at, I think the models that you have, we can look at those questions, like when you add chemo, do you get overexpression of class 1, class 2, PD-1, CTLA, all that stuff, in the tumor cells itself, and in the animal models too.
I wish Dan was here right now.
Yeah, he better.
He's on his
He's running.
That's-
So, leave it pause as a big chemo, or is that something you know? Or
So-
I don't hear anybody talking about leave it.
Yeah.
Which is what was offered to me
Yes.
multiple times. I was
Yes.
just wondering where that falls.
Well, we have this trial now, which we didn't have a month ago, so.
Where does leave it fall into?
Yeah, no, it's a very good question.
Anybody taking leave it and offered leave it as an option?
A lot of people.
Crickets?
Yeah, my husband have chemo.
Oh, yeah, so we discussed it, yeah. And no work, huh?
So all those drugs that we've heard about, Gleevec, Tagrisso, or afatinib, or everolimus, rapamycin, and a bunch of others, there's various studies over the years that showed activity, but it's on the low range of less than 10% objective response rate. And I think this is a very heterogeneous disease. It's very everyone's different. Some tumors are extremely slow growing. Some tumors are very fast growing. So this is why it's very hard to study it. And there are certainly new drugs too, and new approaches, new targets to go after. Ultimately, I think you need combinations. I don't think Gleevec alone, Pemetrexed alone has reasonable activity, better than the others, but obviously we can do better than that, and we want to do better than that. And we will get to combinations, strategic combinations in the future. So.
Have you thought about any ADC to treat chordoma? What was it?
Have you thought about any ADCs, antibody drug conjugates? So these are antibodies that have a chemo linked to them. So it's a great question, and so I do a lot of profiling of the tumor tissues using multiple different platforms, like DNA, RNA, but also proteomics. And I have 2 glioblastoma patients that have TROP2 expression, which is this breast cancer drug, a bladder cancer drug, Trodelvy, and they responded to it. It took me months to get it approved. I don't know whether that's an ADC. I don't know whether it's expressed in chordoma, so I'll have to look into that. But there is more, besides RNA, DNA that we all do these days, there's a lot of proteomic expression that we need to do, because all the ADCs have to have a protein target.
Yes, what's the target for that? The drug?
It's called TROP2.
What's the antigen, the name of the antigen?
TROP2.
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 Oh, TROP2, okay.
TROP2. TROP2, yeah. It's expressed in triple negative breast cancer, bladder cancer, and then when I screened a whole bunch of cancer patients using a company called mProbe, we can see a small percentage of other tumors also express it. So I'll have to look to see whether that's possible. HER2, the breast cancer marker, I don't know the expression, but that's a very good question because there's a great drug called Enhertu that's approved for breast, but now it's approved for lung, and any cancer that has HER2
So my
could
husband-
respond.
Actually, my husband used HER2 for 2 doses, but it's very toxic because the DAR8, it's very toxic. So he had to drop out of it.
Yeah. It is, even though it's targeted chemo, and sometimes it could still have the same side effects.
Yeah.
But tucatinib or the tyrosine kinase inhibitors may be useful to try.
It's hard to say.
Yes?
I've got an easy question. It's kind of basic, broad cancer. In our lifetimes, in our kids' lifetimes, do you anticipate the first line of treatment being other than chemo or surgery or radiation?
I think what's happening in all of oncology, and I have a proposal for the Chordoma Foundation to think about, is to do neoadjuvant. So before surgery, so if we had, I don't know, how clearly can you tell it's a chordoma and not another tumor on imaging? I'm gonna ask the surgeon because
I would say not so clearly
Yeah.
that I would forego a biopsy.
Right. So one approach is to do a biopsy. In other situations, sometimes, in Japan, for glioblastoma, the other main tumor I deal with, based on the imaging, all the fancy imaging, they said this is a glioblastoma. They didn't do a biopsy, and they gave the patient chemo, Temodar, Avastin. 80% of the tumor shrank. And then the surgeon a month later was able to take it out easier and better and safely than before, because there's a lot of side effects and symptoms those patients get. But an approach for chordoma would be to do a biopsy, or if we get the blood test of working, maybe that will help us. And then you do immunotherapy, or Pemetrexed, and Keytruda, for instance, or just Pemetrexed, since we know that works. And maybe they'll shrink it enough such that you can get a better margin, because one of the theories of why it comes back is there's microscopic disease in other areas. So that's one approach. So I think that's where all of cancer is going towards in the future.
Thank you.
So what about bispecific antibody? Have you ever thought about it?
I think about it, but I don't know what. But it's a good project. We need to define the targets for chordoma on the surface, like
Yeah.
TROP2, HER2, and everything, and all
Yeah,
that stuff.
so now there
But the T cell engagers, bringing T cells there, great idea, and I think we could propose that as well.
So as oncologists, it's hard to, for patients, you order, IHC, I think, if you want to find a new target, you've got to do, say, you think about some tumor antigens, you've got to order, some special test, right, special IHC. But right now, you say, for example, UCLA pathology cannot do a lot of stuff, it's very hard for patients. I know doctors just blindly try, this chemo agent, that drug, at the end, the patient suffers so much and
Yeah.
no efficacy.
Yeah. I don't do that. I do order, and it's not UCLA lab. I order all the companies that are external. Caris, Foundation Medicine, Tempus, OncoOmics. There's like 50 companies out there that, and sometimes they are very good and don't charge, they try to build insurance and get reimbursed, but I never order if they bother my patients. So usually I get it done for free for the most part. But we can do it more organized way, is what I'm hearing, for all patients, and we could talk about, like, every patient with a chordoma, making sure they have, good surgery, good treatment options, radiation, chemo, clinical trials, but also did they get the best profiling? Even in lung cancer these days, what you just said, there are in some small areas or community places that they're just giving chemo and didn't even check for EGFR mutations, because the standard is EGFR directed therapy. So that still happens today. And so, you know, we could, actually, we were supposed to do a white paper last year after the Chordoma I thought we should have done a white paper last year.
On,
After the meeting, the Chordoma Foundation meeting.
the research workshop?
To talk about clinical, like, I was doing every 6 weeks imaging, and now I'm doing every 12 weeks, every 9 weeks, 3 cycles. So, but then other people are doing different, the
Right.
cycle length was not the same. So we should have a white paper where we talk about clinical trials, what to collect, and a standardized approach in Europe and in the US. Yeah.
So I have a question outside of clinical trials, if a patient can really do maybe just one type of profiling test, is there one that you would recommend over others?
I've been a big user of Caris for a long time because they also publish with me. They will allow us to ask questions and help us publish data.
Okay.
We have a Chordoma paper with them that we're trying to publish, and they contributed to our paper, the first paper that we did. So it depends on the openness of the company. But then the proteomics, which I'm gonna look at the Chordoma patients and what I've ordered on those, the proteomics company, but there are other companies that could be helpful too. We could talk about that.
Can I ask you a question?
Yes.
If we've had that profiling done, is there some repository that collects this that we could send it to? Does the foundation have it, or does somebody keep all of this so they have all of the?
That's a great idea. Does the foundation have it?
We don't have a registry ourselves, but the natural history study that the National Cancer Institute is doing, their rare disease network, that Chordoma is one of the natural history study of rare tumors. If you go through that natural history study, they'll collect all that from you. They do some of that themselves too for everyone who participates. But basically you're just sending them all of your medical records, and that would include those tests if you've had them, and they may do some of their own things, but that's the main one that's going on right now.
And do you guys have access to that data, or is that just for them?
They're making it accessible whenever they can. We don't have free access to it, like where we can go in and query things and report on it, things like that. But they're doing a lot of that, and if there's really important questions that we have that they may be able to help with, we can work with them to look at the data to get those answers.
So would it be helpful if we sent you our studies so that you have a bigger pool to glean information from?
Absolutely. I mean, we're going to talk about, there is another program through a company called XCures that we've set up for brain tumors and pediatric brain tumors, and we've discussed it with the Chordoma Foundation as well, which is actually very easy to do as well, to get all your data. And so we'll figure something out. We need to do that.
I've got a study, it just sits on my computer, but the only one's ever looked at it is me.
And what did you
And I don't have any idea.
Well, it's interesting, like, everything's becoming AI now. So all that data, we can really crunch it. And
ChatGPT and tell me what
yeah,
this
yeah,
is
yeah.
gonna look like.
You mentioned AI. I am curious as to how you see AI really enhancing the work that you're doing.
Yeah, I think it's gonna make me less efficient initially, right? Just like all EMR medical things do, and make the doctor do more work because I can't ask my nurse to help and other people to help. I have to sign every little thing. So anyway, that was the negative side of things. But yeah, it will help us because, we see, 20 patients a day or whatever. We have ideas, this is in the real world, not outside of clinical trials. That's the other big thing, right? We are treating 1 million cancer patients in the US every single year. 95% are not on a clinical trial. But it's real world data. And that's what I'm working with XCures on, is we collected over 1,000 glioblastomas around the country, just by patients going online and just signing up, and that's all we need because all the records are electronic. So from the Health Information Exchange, we pulled all the records. And in this abstract that we presented last year, it was only like 500-600 glioblastomas. About 300-400 patients got Pembro, or checkpoint inhibitors off-label. And we were able to see, even though the drug failed in the clinical trials, in the real-world setting, patients who got Pembro did better than patients who didn't get Pembro. And we accounted statistically for all the other variables, age, size, all this other stuff. So in the real world, in these large data sets, we can learn a lot. And in the Chordoma, because it's such a rare thing, 200 to 300 a year, if we can collect all that data and use AI statistics, so we will learn a lot about all the little things that everyone does and whether they respond or not, what it is. But we need the molecular data too, and we can gather all that and understand it. So yeah, I think AI or software or data analysis is going to be very important in the future.
So if you have a Chordoma patient, what kind of test would you order in terms of molecular biology?
So I do KRAS on every single patient.
What is
And,
that?
it's just an external, so most hospital testing, they do a panel of genes, like 40 or 50 or 100 genes.
Microarray?
It's NGS usually.
Yes, okay.
Next gen, it's just sequencing of a
Yeah.
bunch of genes. And then, but KRAS and Tempus and these guys also have panels, but KRAS does 22,000 genes, does everything. So 15 years ago, when you had lung cancer, we just did EGFR. That was it, right? If you're EGFR, then you got EGFR inhibitor, otherwise you got chemo. I'm making it very simple. Now, there's 20 other mutations a lung cancer patient could have, so you need to do all 20 of those. But now it's just as easy and cheap just to do all 22,000 genes.
And what about proteomics?
The what?
Proteomics.
Oh, so that's KRAS. And then there's other companies, there's several companies. The one that I mainly use for proteomics is called OncoOmics or MPro.
Okay.
They're in Maryland.
Yeah.
And then a newer company called they changed the name. It's called Ignite Proteomics. They do phosphoproteomics, activated EGFR. I think that's going to be very critical also.
So you also order those?
I do. So most of them don't cost anything or built insurance. The Ignite Proteomics, they do charge patients about $1,000 because it's only covered for breast cancer right now.
Okay. So once you get, say, the sequencing data, who's going to interpret?
I interpret it, yeah.
You interpret?
Yeah.
And read it?
I use my natural AI to interpret. And I look at the literature, PubMed, look at the publication, it's a lot of tedious work. It depends on the target. Sometimes it's easy, sometimes it's hard.
Yeah, those are just common targets, right?
Yeah, but sometimes it's complicated. Even at the NGS,
Yeah.
like some patients have 3 or 4 mutations. What is that? Which one do you go after?
Right.
Which combinations do you do? So it can be complicated in certain situations. So. Yes.
Can you speak about?
God, this is harder than a scientific.
Your information about what you think about monoclonal antibodies, either like specific that would target chordoma cancer cells, either for a diagnosis and or possibly treatment that is, just specific for that cancer.
Yeah, so that's where her question about protein expression on the surface, HER2, TROP2. So we would have to look into it, but I'm sure we could either use something that's already there. Most likely, it's probably not one thing that's, but maybe 80% of chordomas express one thing that we can go after. But it's easy enough to do these days, figure that out and figure out what to go after. And if it's already available, then we could propose a new trial that the Chordoma Foundation will fund. And it would be great to combine that with Keytruda checkpoints, or ideally a chemo, a Keytruda, and something else can really, give a huge response rate is what we want to do. But we have to build it one at a time, to get there. So Erbitux is a EGFR antibody, right? The one that's in trials right now. So.
Thank you very much.
Okay.
Thank you.